Hello, I'm Chris Todd. Alex Jeffries and I are going to tell you about why DNA mutations are important in cancer. DNA is our genetic material, and it's changes in DNA that can lead to cancer. Hence, this means cancer is a genetic disease. However, rare for our reproductive cells to be affected. It is mostly our somatic cells, the main cells of our body, that become affected. We're going to look at this in some more detail. Most of our cells contain a full set of instructions. A full set of DNA is termed the genome. Our genome is like an encyclopedia. Each encyclopedia has multiple volumes, just like we have separate chromosomes. And within those volumes, there are separate entities. In the genome case, those are our genes. But just like in the book, the order of the genes is also important. Not all of the pieces of information, so not all of the genes, are being read in every cell. The difference is the so-called expression profile makes cells different from each other. Within our cells, the genes are being read into those molecules that actually do things within the cell. Most of those doing molecules are proteins. Which proteins, how much of those proteins, and at what time they're present determines exactly what the cell is doing. The patterns of expression of proteins are very complex and dynamic, ever-changing. Some of the complexity of the expression profiles has been worked out for some cell types, but this still remains to be a huge challenge for those researching cancer. Some changes or perturbations to what's going on in the cell, the genes that are being read, can be tolerated. However, too much perturbation can lead to a cell's death, or it can lead to the cell behaving abnormally, i.e. becoming cancerous. Mutations in the DNA are one of these types of perturbations.
Thanks, Chris. So let's talk about mutations in more depth. And in order to do that, let's take a little refresher about DNA replication. So you'll remember that DNA is this double-stranded, double helical structure. The two strands split apart, and these are what we would call parental strands. They also function as templates, each for the synthesis of a complementary copy of DNA. So once these two strands are separated, new copies of DNA are made. Chromosomes can be segregated out into two different daughter cells, and that's how cells propagate themselves. But during this replication process, errors can occur. Firstly, in the actual replication of the template strand, the DNA polymerase can sometimes make an error and put in the wrong base or skip bases, etc. Alternatively, the replication machinery may faithfully make a copy of the template strand, but the template strand itself has got a mutation in it, due to, for instance, exposure to chemical agents or radiation. And lastly, there's a class of mutations that can occur to DNA which isn't strictly part of the DNA replication process. It's part of the chromosomes, how they line up with each other and segregate into the two daughter cells. And during this period, you can get recombination events where sections of one chromosome gets spliced into another section of another chromosome. Now, mutations are not desirable things in cells, so cells have evolved numerous error correction mechanisms. And the first line of defense, as it were, against this is the actual replication machinery itself. So the DNA polymerase, as it is running along, has a proofreading mechanism where it can check that what it's put in is correct. And if it detects an error, it takes a step back and it tries to correct that error. At the same time, there's a host of other DNA repair enzymes that are constantly monitoring the DNA inside a cell, trying to detect mutations, and if it sees them, they try to then correct them to the right form. Now, despite all these repair mechanisms inside cells, sometimes mutations get through, and because it's a piece of genetic material, this will subsequently get replicated into all the subsequent daughter cells, and that mutation gets passed on. Now, as you've seen in week 1 in this diagram, normal cells, in this case, one cell has undergone a mutation, and that mutation is now being propagated into all the subsequent daughter cells that divide out from that. So let's look at mutations in a little bit more detail at the molecular level. You can categorize mutations in various ways, and the simplest type of mutation that you can have is what we call a substitution. The example I'm showing here, we have the parental template strand, which has a G in one location, and after the mutational event, the progeny strand has now been substituted, that G, for an A, and that now gets passed on to all the subsequent cells. Another form of mutation is a deletion mutation. So where the template describes some bits of information, some bases, during the mutation event, what happens is some of that information gets lost. The flip side to that is where you have an insertion. So where the template didn't code for something, now the new progeny templates, progeny DNA, has got bits of extra information, extra bases put into it. A type of mutation that involves both deletions and insertions is what we call a translocation. So a section of DNA from one bit of DNA molecule is deleted out of it and moved to another location where it gets inserted. And that event can happen within the same DNA molecule, or it might come out of one chromosome and insert itself into another chromosome. A variant on translocation is where the region that's coming out, as it were, remains there, a copy is made, and the new bit of DNA goes in somewhere else, so we end up duplicating a section of the DNA. And lastly, because DNA is a linear stretch of sequence, and it's the actual sequence itself that gives the information to the DNA molecule, if you flip a section of that DNA over, you've changed the sequence, as in this example here. Now these types of mutations, insertions, deletions, translocations, duplications, inversions, can be relatively small stretches of DNA, a couple of bases, or they can be very large sections, thousands, tens of thousands, even millions of bases. What effect is this going to have on a cell if it has a mutation? So the easiest outcome that you can envisage is where the protein is no longer produced. So the normal cell requires a particular protein. Due to this mutation, that protein is not produced. Alternatively, a protein can be produced, but it's changed its identity to some degree, either slightly or quite significantly. That could then make the protein product non-functional. So it's equivalent to the first example here, a loss of function mutation. Or the new protein could actually have a slightly different function, which is different to what it should be. Mutations can also increase the concentrations of proteins, so make more protein or less protein than what it should produce under normal conditions. Mutations can also change the timing in which proteins are produced, either before or after their normal time point of production. And there can be knock-on effects here. This is all interrelated. So you can imagine a protein that is no longer produced that has a knock-on effect on other proteins that haven't actually got mutations themselves, but these proteins are now affected, changing their timing or levels of expression or some other characteristic. What effect this has on the cell can be varied. The cell might shrug this off and consider it insignificant and nothing really happens to the form or function of the cell. Alternatively, the cell could actually sicken and die. What's of most interest to us when studying cancer are the cells that remain viable and still continue to replicate, but their form and or function has changed in some way to an aberrant function. One example of aberrant cellular function is cancer, where we have uncontrolled cell growth, proliferation and spreading. Now, cancer is going to arise rarely through a single mutation in a single gene. Typically what happens with cancers is multiple genes in the order of up to 10 or so genes all get mutated. And it's only when these specific subset of genes are mutated that the cell might become cancerous. There are particular genes that are more prone to producing cancers, for instance DNA repair enzymes, as you might expect. And because it's cumulative genes that have to be mutated, it means that the longer a cell line or a lineage of cells is subjected to mutations, simply through replicating itself or exposure to chemical agents or radiation or some such, probabilistically you increase the chance that you'll get sufficient mutations so that the cell line becomes a cancer. And that's shown here in this diagram, where we've got an accumulation of multiple mutation events, and at some point, let's say arbitrarily here at the end point, the right combination of genes are mutated and you end up now with cancer cells.
Thank you for that, Alex. So we've seen that mutations do matter in cancer. Some cancer research is looking at DNA mutations, attempting to identify those common themes and the differences between different sorts of cancers. They're also looking to identify those mutations that are important in the diagnosis or the prediction of cancer. There already exist examples of treatments that have come out from our studies of mutations. And we're hoping the continuation of this research in the future will help develop even better treatments and new drugs.